Announcement before we start the first session of the day. So today we're going to hear talks by Ben Domingue and Dalton Conley. We're going to hear Peter Visscher's keynote, and then we'll have a 1-hour discussion that's off camera, just like last year, and that'll be an opportunity to raise really any issues that are on your mind and that relate to the themes of this conference. If you have something you'd like to say, we can always continue the spirited conversation from yesterday. If you have other topics you'd like to raise, just feel free to approach me during break, and I'll make sure you have an opportunity to speak up early. I think that's all I want to say. And with that, I'll hand over to Ben, who's going to give today's first talk.
Good morning. So I'm going to talk about schools as moderators of genetic associations with Lifecourse attainments. And the things I'm going to talk about are largely in this published paper that came out this summer, so if you'd like to see any of the details, you can, of course, refer to that. So the motivating idea here is that genetic influences need not be constant across environment, and in fact, if we were to observe consistent and strong gene-by-interaction effects, we would need to be kind of careful in interpreting genetic main effects since they would be environmentally contingent. And so the key question we're going to pose in this paper is whether school assignment needs to be accounted for in models linking genotype and attainment. In undertaking this kind of G by E work, however, I think it's important to keep in mind the sins of the past, as it were. So there's an entire decade of candidate gene by environment work that has not really withstood the test of time. And so what we're going to be doing, I think, needs to be informed by the failures of that previous era. And in particular, I think the review by Duncan and Keller is very helpful in illustrating the things one ought not do again. So in thinking about schools, I think there are two things that are worth kind of pointing out right up front. So first, schools are an educational environment we clearly care about, right? They're a public policy intervention of the kind of highest order, right? We spend a massive amount of money and time trying to educate kids in a responsible way, and yet it's also been incredibly challenging to pinpoint the specific environments that's, of the many environments that you can choose to measure in a school, it's proven hard to figure out which ones are actually relevant. And this kind of observation goes back to James Coleman and some early work that he did. And so I think the question this raises for me is which specific environment do we care about in posing these kinds of G by E questions? And so this is going to influence our work in the sense that we're going to be, I think, kind of attentive to the fact that we don't really know up front which environment is the right one. So the first thing we're going to do is just look in a kind of indirect way at whether associations between polygenic scores and attainments differ across schools. And then we're going to choose a few candidate environments that we're going to specifically measure and ask whether these specific measurable environments moderate the observed association between genes and attainments. So we're going to do this work using two longitudinal data sets. They were both discussed yesterday. So the first is AdHealth, and this is a data set that contains about 5,000 individuals clustered in about 137 schools. And then we're going to use the Wisconsin longitudinal study as well. And this is nearly 9,000 individuals in over 400 schools. There's also the separate problem that the Wisconsin respondents come from families. I'm not going to focus on that, but that is something that we deal with analytically. I want to emphasize that, at least in the US, there aren't a ton of data sets that contain both genetic data and this kind of school-based clustering, and so there aren't a ton of opportunities to do this work. These are two of the only data sets that would allow for this kind of investigation. However, they're also very different in the respect that they cover very different birth cohorts. So the AdHealth respondents were in secondary school in the mid-90s. The WLS respondents were in secondary school in the mid-50s. And this is going to have implications for our results, as you'll see. So from these data sets, we're going to focus on a few things. So one thing we're going to focus on is the individual's polygenic score. And we use the EA3 scores constructed by Robbie Wiedow. And then we're going to look at two types of attainment. First, just years of education, how long somebody's in school. And then second, an individual's job status. I'm not going to talk as much today about this occupational measure. If you're interested in that, we have this separate paper, first authored by Dan Belsky, that covers the occupational stuff in great detail, so you could certainly look there if you were wondering about that measure. I did want to emphasize one thing about the educational attainment measure. Here are the distributions. On the top we have AdHealth, on the bottom we have WLS. And what you will notice especially about WLS is there's this real kind of heaping right there at 12 years of education. And this is largely a function of the way the WLS was designed. You had to be a high school graduate to be a part of WLS unless you were a sibling of a high school graduate. And so that leads to some complexity, and we deal with that in the paper, but in the talk I'm largely going to focus on ad health just because that complexity is, I think, distracting from the main points that I want to talk about. So here are two analytic approaches. So the first is the indirect approach that doesn't assume any knowledge of the specific environment that's relevant, in terms of moderating this genotype-phenotype association. And we're just going to use a random effects model where we have slopes and intercepts. And so I've highlighted the kind of slopes and intercept part. And the key question here is whether we observe variation in the delta J's, right? And so do we observe some kind of substantial variation in the slope linking genotype to phenotype across schools? The direct approach is where we're going to use some specific candidate environmental moderators, and here we're just going to rely upon standard linear models where we include interactions. That's the lower red box. And the question is just whether the beta 3 term is big in some sense. Okay, so two caveats. So first, there are some power limitations here. I don't want to get too far into the details about power, but I do want to acknowledge up front that we thought about power. In particular, we thought about how power would be affected by measurement error in our measure of the environment, because given that we don't know what the right environment is, we might have some ideas about proxies that could capture some of the features we care about. That's not the same as having the exact environment, so we thought we should worry about the role of measurement error in our environment, and that goes to further attenuate power. So we have some power limitations. We also don't have exogenous kind of environments here. There's no sense in which in the US your school is exogenously assigned to you, right? There's a substantial amount of evidence that suggests a great number of things play a role in tracking you into different schools. In particular, we can see, especially in ad health, a very strong association between a measure of school status, in particular the educational attainment of the parents whose kids are in the school tracks strongly with the mean polygenic score of the students in the school, right? So we don't have exogenous assignment either. And these are both important caveats that we talk about in the paper, but I do think that the key messages that I'll try to emphasize today won't overextend and overstep what we can say given these limitations. Okay, so here's the first result. So what we observe in this panel, so this is based on our random effects models where the x-axis is just the polygenic score and the y-axis is educational attainment. I've emphasized some of the most extreme slopes with the red lines. The gray lines are some of the other schools. And what you can generally see is there's just not a huge amount of evidence here for substantial variation in the slopes of those lines. They all look pretty similar. And we can test this via randomization analysis. So what we did is we went back and we randomly grouped students into kind of quote-unquote schools. And when we did that and then re-estimated our models, in about 20% of the cases, the random assignment to school yielded just as much or more variation in those slopes, right? So from this perspective, we're really not able to observe much in the way of substantial variation in the slope of this genotype-phenotype relationship. But what there is very clearly is variation in the intercepts, right? You can see that those lines all look parallel, but they obviously have very different kind of intercepts on the y-axis. And this is very important. So the predicted attainment, right, for a fixed genotype varies quite a bit as a function of which school you go to. So the different distributions here are distributions of the number of years of education we would expect for a fixed polygenic score. And what you can see is very substantial variation there. In fact, the variation there is much bigger than we would expect just based on the variation in the polygenic scores. In WLS, it's a little more complicated. You can see in the red distribution at the bottom, those are the lowest scored students. They have fairly limited variation, but as you kind of move up the polygenic score scale, you can see that kind of distribution widening out. In ad health, it's a little more, those distributions are kind of uniformly variable, but in both cases, I think there's very substantial variation in the intercepts, as it were, right, in the predicted number of years of schooling for a fixed polygenic score. And this is going to have two consequences for us. So the first, it really kind of causes some problems in our random effect analysis, and so I'll show you a little bit about how we tried to resolve that. And then it's also going to have very substantial implications for how we think about analyses of dichotomous outcomes. In particular, how we think about credentialing type analyses, so analyses of whether or not you went to college or completed college. So in terms of getting around this problem of the substantial variation in the intercepts, the first thing we did is we reran our randomization analysis where we mean center years of education within school, and that doesn't really change the overall result too much. We still see that in about 6% of the cases, random assignment to school yields just as much variation in the slopes or more than the true school assignment, right? So even when we kind of account for the substantial variation in the intercepts, we still don't get strong evidence of G by E here. Now what I want to do is turn to a dichotomous indicator, and so I'm going to look specifically at whether you attended any school past high school, right? So whether you attended anything in the post-secondary realm, and roughly 75% of the ad health respondents were in some formal education past secondary school, right? So this was a fairly common outcome for the ad health respondents. And what you can see in that right-hand panel is a picture that shows what looks like at first glance a much more substantial variation in the slopes, right? I think it's fairly obvious that those lines at the top have fairly flat slopes. As you kind of go down, those lines at the bottom well, the slopes are much steeper, right? And so I want to kind of try to tease apart what's going on here. So first I'll note that a randomization test of the kinds I've been talking about does support the inference that the slopes really do vary in that right-hand panel, right? So that is a true thing. But what we're going to need to be is very careful in terms of how we interpret that. So I want to kind of draw this out via a simple cartoon. So here are two regression lines based on simulation wherein the true relationship between the variable on the x-axis and the variable on the y-axis are in fact parallel, and those lines look pretty parallel. So when we think about a dichotomization, right, in this case I'm going to dichotomize via that blue line. What we're really doing when we dichotomize is we just focus on the proportion of the conditional distribution above the blue line. And you can see for the black line, almost all of the conditional distribution is above that blue line, and for the red line, well, you know, maybe something like half of that distribution is above the blue line. And if you think about kind of sliding that vertical bar across the x-axis, that would create the kinds of curves that tell us about the relationship between the x-axis and the probability of being above the blue line on the y-axis. And the punchline here is just that if what you in fact have are these two curves, you're really going to be struggling to identify the fact that they are in fact parallel in some sense, that they come from the parallel lines on the left, especially when you have kind of different distributions for the two curves to begin with, right? So the black and the red distributions in that second panel are meant to reflect the fact that we have kind of shifted distributions in the different schools as well. And so if I gave you a set of draws from that black distribution and a set of draws from that red distribution, I think you'd be really hard-pressed to identify the fact that those red and black curves were kind of parallel in the sense that they came from the same parallel lines on the left-hand side, right? So I think it's going to be very easy to misinterpret variation in intercepts as variation in slopes when we have dichotomous outcomes and we aren't kind of thinking clearly about what those dichotomous outcomes are meant to tell us. I'll show you more pictures along these lines in a moment. So now I want to talk about the specific measured environments we use. And so the first measured environment is just going to be a measure that's kind of meant to capture school status. And so it's just the percent of moms for the respondents that have a high school diploma. And this is what I've been thinking about as a structural environment. And by that I mean it's an environment that we expect a priori to have a large environmental effect, right? We expect a big E here. In contrast, we also considered a measure of school stratification, and this was a measure about the inequality in parents' education, right? We computed a Gini coefficient for the distribution of parental educations in the schools in our sample. And I'm still trying to think of the right word here, so I'm open to suggestions. I'm calling this for the moment a differentiating environment. And this is an environment that has an ambiguous main effect, but there's some theoretical rationale for believing that G might have varying implications as a function of the environment. So if you go to a school where educational outcomes are very, evenly distributed, that might have implications for how the school sets up curricula. Maybe there's less tracking, right? Maybe there's kind of more of an effort to help all students achieve at some level. these are all just kind of hypotheses, but at least I think up front it's plausible that you might have environments that are really going to differentiate how kids are treated as a function of their endowments in these types, across these environments. And so a few more notes about this. So both of these environmental measures are constructed with relatively complete student bodies, not just the genotyped respondents. So as Kathy mentioned yesterday, the AdHealth students, all of the students in all of the schools were interviewed in a briefer interview at Wave 1, and so we used that data to construct those environmental measures. And we also, in our analyses, use the saturated models that Matt Keller suggested in a very useful 24 paper in terms of thinking about how to do G by E analyses. So I'm going to show you a bunch of pictures that look kind of like this, and I just wanted to orient you towards what we have here. So on the x-axis, we have just the value of the polygenic score. On the y-axis, we have some outcome. This is educational attainment. I'm also going to show you the occupational attainment results. I've come to think that it's probably much more important than I previously thought to pay attention to the distribution of the outcome. And so the thing on the y-axis in gray at the far right, that's just the distribution of the outcome. And so we can kind of see where the stuff is happening in terms of the lines, in terms of where the people are via that distribution on the right. The red and blue lines represent kids who are in very high status schools, that's the blue line, and kids in very low status schools, that's the red line. And so what we can see in this first analysis is that in AdHealth, those two lines look very parallel, and in fact the kind of inferential tests at the bottom suggest that they're pretty much parallel, right? So this again tells us that in AdHealth, there is little evidence to think that being in a higher status school does much more than just bump up the intercept of the line linking genotype to phenotype. So here's a bunch more pictures. So the first row is educational attainment. The second row is occupational attainment. You can see it has a very different distribution than the educational attainment one. The first column is the school status variable I talked about. That's the percent of moms in the school that completed high school. The second column is the stratification variable. And in general, we have kind of null results. So in the bottom row, everything is null. In that top left-hand panel, things are null. We have one potentially interesting result here showing that there's some evidence potentially for stratification such that kids in more weakly stratified schools have flatter kind of associations linking genotype to phenotype. And so the first question to ask here is, does this replicate in the WLS? Because if it did, that might start to be kind of interesting. It in fact does not. Not only does it not replicate, but the sign of the relevant thing is in the opposite direction. So I don't know that we want to get too excited about the AdHealth result. But what I do think is interesting is this result, right? So what we have here is evidence to suggest that WLS respondents in low status schools, that's the red line, had limited opportunities to pursue higher education, right? So we can see that those students in the red line, right, irrespective of what their PGS was, well, not irrespective, but even for high PGS students in the low status schools, they did not have the same opportunity to go to higher ed as the students in the blue schools, whereas we see kind of relatively similar types of behavior for low PGS students irrespective of school. And I think this is potentially consistent with other scenarios wherein structural constraints limit variation in some outcome and thereby induce a gene by environment interaction finding. And so I've listed a number of papers here, several of which I'm sure are near and dear to the audience. And I think this is the type of finding we're likely to see quite frequently, and I'll talk a little bit more later about what I think it means, but it's caused me, at least personally, to wonder what it is we're trying to do in this type of analysis. And I do think it's worth emphasizing that we don't see that G by E finding at the top carrying over to the bottom, right? So just because kids in the higher status schools that had high PGS scores were more likely to go to higher education, that didn't translate into them also being more likely to have higher, we don't see then a break in the slope when we think about occupational attainment. So I think that limits the extent to which we think this is like a structural finding of great interest. So now let's look at the dichotomous outcomes, and I think the kind of big point here is just with these dichotomous outcomes, I think we need to maybe be a little more cautious. And in some ways, this is probably me criticizing myself since I've done work with dichotomous outcomes before, so I think I've learned something here as well. So I have two types of panels here. On the left, I have some descriptive curves. The densities are the densities in high and low status schools. Notice that they're kind of shifted from each other, and that is meant to emphasize the fact that you have kind of different types of observations across the two schools. As I showed you in that picture earlier, right, where I had the blue, where I had the black and red lines shifted. And on the right, we see kind of, a big G by E finding in the sense that we have seemingly very different associations between the chance of going to post-secondary school and the school status. But in fact, this is all E, right? This is all due to environmental effects. We have kind of parallel lines in ad health as a function when we think about genotype predicting years of educational attainment, and that does translate into these different probabilities of attending post-secondary school, post-secondary schooling across schools, but this is not due to a G by E effect. This is just all E that we can misinterpret as G by E if we're not careful. And I think this point has been made by others. I'm going to kind of skip the WLS results just out of time considerations, and let me just say a few things before I leave. So one, I think we have relatively limited evidence for moderation of the PGS attainment relationship, especially for thinking about variation in slopes and not intercepts. The second is that the moderations we do observe are consistent with a structural interpretation, wherein environments are really molding outcome distributions. And so this has left me thinking three things. So first, I think we often would be maybe better thinking about generic environments rather than very specific environments, given that I don't know that we have the best ideas about the specific environments that are going to induce G by E. We know which environments have big main effects. I don't know that those are necessarily the environments that should also therefore have G by E effects as well. Second, the variation in intercepts may be useful in terms of identifying environmental effects, but I think they're going to pose challenges in other situations, such as when we want to study dichotomized outcomes. And finally, I think G by E findings driven by structural constraints on outcomes are certainly not the thing that were initially of interest to people pushing gene by environment work. I think this is a kind of a horse of a different color, and in many cases, I think we're just kind of rediscovering the fact that environments have big effects. So this is the paper if you're interested. I want to especially thank Sam Trejo, who is in the room, I believe, who was instrumental in getting this work done, and I want to thank the funders as well. Thank you.
This follows very nicely on Ben's because it's another G by E paper. It's very unlike Ben's, and it couldn't be more opposite in the sense that he's presenting a published work. I'm showing you stuff that literally I was up at 3 a.m. last night working on, and my poor RA's back in, it's 3 hours later there. So many thanks to Rebecca Johnson, who's leading this, Romina Sotude, a graduate of the SSGAC training session a couple years ago, and myself. So I think I don't need to tell, I know they're a mixed crowd, but I don't need to tell the social scientists in the room why we think G by E is important. Often, so maybe I'm speaking more to the geneticists now, why we care about this. Almost all across the domain of social sciences, we find heterogeneous treatment effects. Even when you do a randomized controlled trial, if you end up with a 10% treatment effect, that would be, for most of our outcomes, enormous and amazing, especially in education research. But if you look at the data closely, you find that there's a huge heterogeneity in the response to almost any treatment, in that 10% is probably driven by 10% of the kids doubling their reading scores rather than a 10% across the board average treatment effect. And the intuition for many of us social scientists that went into this was that perhaps pre-existing biological disposition and genotype in particular might be the magic Pink Floyd prism that would take this white light of an average treatment effect and refract it into the rainbow of colors that would tell us which kids are going to respond to a treatment and which kids, in a positive way, which kids are not going to have a response and which kids are going to have a negative response. But Okay, I'm missing a slide, but okay. So there's a lot of presenting problems. You heard about some of them from Ben in doing G by E work. The one that I focused on mostly up till now is the lack of exogeneity in most of our environmental measures. This is really important because, especially with a noisy measure of genotype, the very genotype that the underlying true genotype that we're concerned with and want to interact our environment with, is often leading us to select into those very environments that we want to see if they are moderators. So the solution to that, of course, is well known in econometrics, using regression discontinuity, ideally a randomized controlled trial, or some other natural experiment where you can plausibly argue that there is quasi-random assignment of the environment. We also need, of course, exogeneity of genetics. We know we talk a lot about population structure. Of course, the recent paper that Peter was, I think, the senior author on, that used 100 PCs and still found a polygenic score distribution across the UK suggests that there is, so even when you control for deep population structure, the very genotype that you're interested in is not randomly distributed across environments, which if you're using it as an estimation, if you're using that distribution of people to do discovery, you really are back to the problem where you can't separate out whether you're discovering genetic effects, putting aside genetic nurturance, or environmental effects. And of course, Alex's approach and others using sibling fixed effects and others might help get us a purer measure of G that when we want to interact with E. Then the third issue that I'm going to talk about mostly today is what do we mean when we talk about G? Most gene-environment interaction work, in fact, I think all of it that's used polygenic approach has used the polygenic scores for BMI, for education, et cetera, that are trained on predicting levels of the phenotype. And especially when we use these polygenic scores that have been estimated through a meta-analysis across a lot of different environments, to take, for example, the Giant Consortium or SSGAC's educational attainment analysis, we are literally estimating the effect on a level that is robust across most of the measured environments that we care about, from social and democratic, very recent cohorts to cohorts from the United States in the first half of the 20th century. So we're essentially recognizing a signal, genetic signal that is robust across all those environments. So we might be shooting ourselves in the foot in terms of actually then trying to discover how is that signal moderated by some measured environment or natural experiment. So an alternative would be to measure, first, earlier work, and this is work by Lauren Schmitz and myself, try to deal with the E side, use natural experiments, in this case, the Vietnam Draft Lottery, which literally was an experiment with a drawing. Its 50th anniversary is coming up next year in December, around this time. And we estimate, we use that as our E, and we looked at smoking cigarettes per day over the lifetime, and we found suggestive evidence of a gene-environment interaction, even using the sort of levels polygenic score for predicting smoking, that the exposure to the Vietnam era military service, which is a very tobaccophilic environment, led you to smoke more for the rest of your life if you were genotypically more prone to smoke, to get addicted to nicotine, is our interpretation. Another example provided by Pat Turley and others used the schooling reforms in the UK, and this has, I think, bunch of papers, right? But this particular one looks at the equalizing effect of schooling across polygenic risk for BMI, both the BMI polygenic score and the educational attainment polygenic score, which has an effect on BMI. And you can see that there's a discontinuity such that for cohorts that went to school in the post-reform era, and the increased education attenuated the genetic differences between individuals. And jump in here anytime if I say something wrong about any of your papers, because a bunch of you are going to get tapped on here. So not only is G by E and the idea of getting the right genetic architecture for measuring it important for social scientists, it's also important for population genetics or even for geneticists writ large. There's a concept called a phenotypic capacitor. Most famous one is heat shock protein 90 that was shown in fruit flies, where if this is disabled through heat shock or through mutation, then it's not that it changes a level of a given phenotype, but it allows a radiation of phenotypes and reveals previously suppressed cryptic genetic variation. And this has been hypothesized as a way to explain Waddington's finding from over 50 years ago of genetic assimilation, that there's a reservoir of neutral mutations accumulating, and then an environment changes or a key mutation to a phenotypic capacitor obtains, and it releases a broad range of phenotypes in a sort of insurance mechanism to hope that one of those phenotypes then is adapted to the new environment. There's a big debate going on right now of whether actually it's de novo mutations that drive this dynamic or not, but you get the general picture that there could be, through epistatic interactions, there could be certain genes that are very important in repressing or allowing variation rather than having an additive effect. And so there's been some work to look at these kind of phenotypic capacitor loci in humans, but in terms of doing G by E polygenic work, the typical approach, as I mentioned, uses the PGS as estimated on phenotypic levels across environments, and today I'm going to talk about alternatives to that. So I'm going to show basically a comparison between three different polygenic scores as interacted with a moderator. And the first one comes from the latest giant analysis, meta-analysis that predicted BMI. That is the levels, and I'm going to call that the M-PGS from here on out, meaning main effects PGS, versus, and again, by the end of this, almost all of you are going to be called out. Peter's other paper from 2011 with Yang et al. Paper used the squared Z-score in what was then the giant consortium with an N of about 133,000 to model phenotypic plasticity, and it used a heterogeneous population, as I was talking about, of cohorts across the Western world of white folks of European descent, and found significant capacitor at the FTO locus. So we have the weights from that analysis, and then we use Alex's recent paper on a well-powered way to look at variance-inducing loci using a heteroscedastic linear mixed model, and that N is about 275,000, and it comes from white British of the UKB. And so I'm going to call that the Yang et al. The VPGS, and I'm going to call Alex's the DPGS, because we're using the weights that come from the component that he estimates for dispersion effects of a given locus. The advance of Alex's approach, and again, feel free to shout out no if I'm saying, but is that he, in this mixed model, he allows for the additive effect for an effect, a sort of global effect of that takes care of the relationship between the mean and the variance, and then allows for the dispersion effects. As it turns out, when you look not maybe at the very top hits, but across the entire genome, the variance weights and the dispersion weights are very highly correlated. In fact, we get the same results I'll show regardless of which we use. You already heard in this meeting, I think, about the HRS, the Health and Retirement Study. So that's our first sample, about 10,000 older Americans, mean age is 57, and the mean education is 12 years, and the mean BMI is 28. And then we're going to try to replicate what we find in Ad Health. And that's, again, about 10,000 individuals, much younger. They're about 40. They're more educated because they're from a younger cohort, but they're reflecting trends in the US. Even though they're 16 years younger, they have the same mean BMI as the older group. And these are both small samples to detect these effects. We'd love to do UKB, and in fact we did it. Everything's significant in UKB using these approaches. Not everything. What we want to show is significant, but of course the problem for that is that Alex's paper used the UKB as its discovery. So these other two data sets, Ad Health and UKB, were in neither the Yang et al. Paper or the Young et al. Paper. And after giving this whole rant about that your environment needs to be exogenous, I'm going to throw that out the window and just use adult measured education as our first pass to test this idea. So here's our first result. We look at the levels PGS or the main effects PGS, and we find no interaction. So the green line is folks whose education is 1 standard deviation above the mean, and the pink is 1 standard deviation below the mean, and predicted BM. Basically, there's no effect, no interaction. When we use the weights from Yang et al. 2011, we get a significant interaction, but it goes in the opposite direction of what we thought. And a couple of you have seen this paper already at IGSS, and this was something I was puzzling over there. So that people with supposedly a higher plasticity score end up not showing an effect of their education on their BMI. When we switch to the dispersion score, we actually find that it's significant and it works in the direction that we think, which is if you have more dispersive alleles, if that's the word, then you see that people of equal education with higher dispersion leads to greater differences. Like Ben, when we tried to replicate this, unfortunately, in Ad Health, nothing significant. So we're going to continue. If we do it in the UKB, where again we have the problem of ascertainment bias because discovery was done there, but we have a great sample size. Not surprisingly, we find that the Yang et al. Is not significant, and the Young is significant and positive in the right direction. So trying to understand where are these differences coming from, and I look forward to hearing from the authors of the original papers, but they're very unrelated, the two measures. And we checked, we thought we had alleles coded differently or strands flipped, and we checked, and at least for now, this is what we seem to have. In fact, if you look at a heat map of the correlations where both the intensity of the dot and the size of the dot represents the correlation between these measures, you can see that the Young et al. VPGS and dispersion PGS are negatively correlated with the levels PGS. and negatively correlated, but very weakly, with the actual phenotype, and negatively correlated with the Yang et al. VPGS. So we're really, different things are going on here, and trying to figure that out. So going forward, we want to redo this using perhaps Patrick's discontinuity in schooling in the UK, or other exogenous environment, of course, because I would argue, again, counter to what I said at the beginning, it's still interesting to look at an endogenous environment in this case, because we are conflating epistasis, the gene-gene interaction, with GE interaction. And as a first pass, that's interesting to see, even if there's any sort of moderation that leads to phenotypic differences. And then later on, dig in to see whether or not it's actually environment or if it's epistasis. So we'll go back to our usual bag of natural experiments, including the Vietnam draft again, birth order, twin differences in birth weight, and so forth. We currently we're using the same UKB sample to run Alex's approach on other phenotypes, and phenotypes of more interest to social scientists, social and behavioral phenotypes, to generate weights for those for DPGSs or VPGSs for those outcomes. And we want to, well, something I could use advice on, again, we have the UKB to do this discovery, the white British, but this is looking at variance. You're even more underpowered than you are looking for main effects. So the trade-off is we have a lot of other cohorts we can add into this analysis, but then we're back to the problem of perhaps not adequately dealing with population stratification. So another idea is to do this with sibling models in two ways: standard sort of sibling or trio analysis for discovery, but also something that I was very excited about a while back, but have since dimmed on, but may look at again, is to use the sibling difference in phenotype as the measure of a variation. So that allows you to basically treat every family as a unit of analysis and separate out mean and variance effects, because you have a mean and a variance for each SIBship, and then model that. And we're doing simulations which show, bizarrely, after everything I just said, that the sibling phenotype method doesn't really work very well in terms of its sensitivity and specificity ratio. And so far, the Young approach doesn't work well either. And actually the squared z-score, the simple approach of that, does the best at discovering true variance affecting loci. So it's right now a big mysterious mess, and we're trying to sort through it all. But thank you.
I'm someone who's from economics, and Dalton's critique of your approach seemed very relevant in terms of that surely you only have a partial observation on the genetics of the kids, and that's correlated with the parents' genetics, and then the parents' genetics is leading them to choose particular school environments. And we know school choice is an enormous element in people's location decisions. And so how do you deal with that problem of confounding?
Well, I don't think we fundamentally do. I mean, this was meant, in my mind, as an exercise to try to understand the degree to which we observe substantial heterogeneity in the returns to one's genotype as a function of school assignment. Deciding what that meant would have been a different endeavor, but seeing that the things are relatively stable is, to me, useful in terms of interpreting, the main effects of the polygenic score. And we don't need to perhaps worried too much about the school environment, at least the kids were exposed to. So I agree entirely that the endogeneity thing is not something we solved, and it is kind of lurking. But I think it, in general, I mean, there's going to be a lot of G by E work that uses endogenous environments. And so I think at least sensitivity to that fact is one thing, and being more comfortable with null findings is another thing that we can, do to at least improve our own thinking on the subject, rather than just confusing ourselves into, finding kind of G by E everywhere we look because we're unattentive to endogeneity or not really focusing on the kind of variation in the outcome variable as we should be.
Dalton was talking about how he could get a lot more power in the UKB, and last night on the way to dinner we were talking about this idea, which is a totally standard thing in other fields. Why can't you just estimate, have the discovery sample be 9/10 of the UKB, then you estimate it on the 10th, and you do that with all the different tenths? It seems like that's a perfectly good procedure to make sure your discovery is done in a different sample. And this probably ought to be routine. And in particular, since it helps you, Dalton, you'd be a good person to start that.
Sure, you could do tenfold cross-validation, but that's not going to increase my N in terms of discovery. That's not gonna,
but it's going to increase your N in terms of...
I mean, what it's going to do is it's going to make the same, like if we think that one of the problems is that there's an incomplete genetic and, in fact, environmental correlation between the discovery and replication samples, which they are, because I'm bringing them across the pond, so to speak, to US samples, that's going to lead to attenuation. So it would solve that because we'd be using the same exact environment and genetic pool. So, but it doesn't, I don't see how it increases.
I thought the problem was the size of the validation sample.
No. The problem is the size of discovery more.
But Ad Health was doing badly, which suggests a problem in the size of validation.
Well, they're both around the same size. No, HRS and Ad Health are around 10,000 each.
Peter?
Just a question for clarification for Dalton and then an update. The methods that use the variances, they can detect in principle G by E without actually having measured E, which is
right.
Probably why we did that in 2012 or whatever it was. Just an update, Yang's group has done this systematically on a whole bunch of traits in the UK Biobank using the full sample, and finds significant individual loci or SNPs, and they're all, I'm looking at Loic as well, I think they're all weight related or BMI related. So this is clearly, there's evidence for G by E. There's also a paper coming out.
But, sorry, how do you know that it's G by E?
Sorry, we don't know for sure, but some of the observations are consistent with actually having identified when you test specific E's like smoking or something else, you can see something like that. Then Oliver Stegel's paper coming out, it's on bioRxiv, but it's coming out in Nature Genetics. It uses a mixed model approach and also finds evidence for G by in the UK Biobank. My question to you is, so you've taken these, as I understand it, the summary statistics, and then you made like a predictor for an individual for their variability.
Plasticity, yeah, or whatever you call it.
Okay. All right, okay. Yeah, no, I'm just trying to get my head around that.
Okay. But I want to ask you guys about that work afterwards.
Oh yeah, we also did in this paper that we're about to put on bioRxiv, we did a systematic comparison of different methods, because as you know very well, all these G by E estimates are dependent on scale. So we try to look at variance-based approaches and try to not have them dependent on a mean variance relationship. And I think the method that is most robust, whereas Alex is something related to the Levine's test and similar to yours as well, I think. So just
well,
that's
I
just
think
a
that's
comment.
Great that now there's like a converging to a method and on that side, and I think it's time for us to join forces and with the social scientists who have thought a lot about how do you think about an exogenous environment to make, to make the claim that you're saying like a smoking locus is one of the interacting locuses
Smoking as an environment.
as a behavior.
Yeah.
That's driven genetically too, so we can't really say that's not GG. So I'm just saying, maybe it is, but sort of, the economists in the room especially have thought a lot about how would you estimate something like that with exogenous exposure. So we'll take if you'll let us have them, that and then test it on the Vietnam exposure, for example, to cigarettes. Yeah. But we can talk. Yeah. This is like, those dreams, at least I have my pants on, but those dreams where like you're presenting something and literally like Marcia McLuhan is behind the, if you know my reference, behind the door. So
I think I just have a little bit of a worry that a lot of what you're seeing is actually driven by the mean effect. So in the sort of squared Z-score approach used in the Yang et al. 2012 paper, that those scores correlate with the mean effects because you still have this relationship between the mean and the variance even after inverse normal transformation. So if you construct a score from those squared Z estimates, that's going to correlate with the mean effects. And then my procedure for estimating dispersion effects requires you to sort of subtract this mean
Right.
effect. So that could induce potentially a negative correlation with the mean, whereas Peter's and Jan Yang's paper would have a positive correlation with the mean. So that could explain why you see this differing direction of effect for those two methods. And then that kind of makes me worry that it's really some sort of mean thing that's going on here and not really a variance interaction thing. But, we should probably have an offline discussion about this.
do you think that it's particularly problematic for using sort of all the SNPs, or is it?
Well,
Because in the original paper, I remember you guys showed the global correlation between effect size for mean and effect size for variance, and I thought that was wrong because the global effect for like 999% or 99.9% of SNPs is zero anyway. So you that induces just a zero correlation. But if you took the top 150 or something like that from your paper, you actually see a very strong correlation. So and you're saying, makes sense what you're saying about your score, that because of the subtraction approach that it would induce negative. But is it more accentuated for the top SNPs?
Well, my guess is that the true distribution of mean effects is much greater than the true distribution of variance effects. So it's likely that any mean signal is going to dominate any true variance signal coming from G by E genome-wide.
But we were interested in SNP discovery, right?
Right.
So we looked at we were very well aware of mean variance relationships, and we tried to do that to death in that paper, and it's also an appendix, supplementary information. And for the particular FTO-related variance, this is obviously related to the mean as well. We even tried to model where there's an unobserved causal variant that is in LD with it, and all that, and to our knowledge, that result still stands.
Well, the fact that you have
My adjustment reduced the FTO variance effect somewhat, but I think it's definitely still there, and I think there are interactions relating to FTO.
and given the different biases, you're saying that kind of should converge on a true effect.
In that second stage, I wonder if in that second stage you shouldn't have the mean effect PGS too. So we've done it. I should have said I've done it with.
We put everything in at the same. We can't put your variance score and your dispersion score in at the same time because they're so correlated. But we do it where we put the main effect, the MPGS, in at the same time. We do MPGS interaction.
What does that
Also,
look like if you put
it's the
put
same.
that in?
That we get the same.
You don't resolve the puzzle.
It's to the extent that it's some confounding on that levels effect that's captured by the weights for the additive effect. It's not that's not driving what we find. I should have said that we test it alone and everything together, and it's still significant.
But you're sort of collapsing it all into one score, so it kind of, yeah, I have to think about that.
Has anybody sort of tried to systematically estimate the heritability of phenotypic variability?
Oh yeah,
Yeah.
that's something I'm working on too. Even though the proposed sibling approach failed in terms of our simulations, it still has this. We still have this idea that if you take if we're brothers and we take our distance and our, from each other, and then we randomly select one of us and tag that family level phenotype to us, then I've got individuals. I can go do Gremel. I can do whatever to estimate the kind of SNP heritability of within family variation, and that's something we're doing as well. That's probably very underpowered too.
Well, just a comment that there are estimates of heritability from other species
Right.
on the essentially the environmental control, sort of genetic control of environmental variants, and typically very small, but significant, like of the order of a few percent or so.
We also looked. We just did kind of very back of the envelope Gremel on your the Yang et al. Measure of squared z-score, and we found for BMI, for example, the heritability was about 1/4 of the heritability for levels. It was not trivial. I mean, it was not zero. So yeah.
Okay, John King, and then we'll take the wood break.
Yeah, on something completely different. But Ben, hi. Dang, I was hoping that was going to work out. But at any rate, one question I have about sort of for the room and stuff is, although I hear that some people at NIH don't think educational attainment is important at all, they are obviously not looking at the data, so I don't care. But seriously, we do have a huge shortage of cohorts in the U.S. where we could profitably look for some of these things. I mean, you've got AdHealth, you've got WLS. In a few years, we'll have some data that people can use from Project Talent. There's possibilities maybe someday getting genetic data from high school and beyond and an LSY79, and I think that's going to be it.
I was going to say the HRS2 is linked with the Lifecourse history module. Might be linking more to school.
Well, no, you'll get something about some of those stuff. I'm talking about looking at school effects.
Right, but
And-
they'll be linking to schools if you.
Right, the 1940 census stuff might help a little bit too. But if there's anybody from anywhere, I don't care, Finland, who cares, anywhere that has access to people, embedded in schools enough genotyping to care about, we should start caring a bit more. Because I mean, I'd hate to say that we know that there's nothing there just based on WLS and AdHealth, because that's not enough. That's a plea for people to pitch us stuff.
Yeah, I think that's fair. I don't think, for a variety of reasons, that our evidence is completely dispositive. But I think it's a useful thing to know that there's not kind of rampant G by E with respect to school assignment, but certainly more work
with respect to high school assignment.
Yeah.
Right, and so this is the other thing too, is that we don't have anything on. It could well have been that it was more sensitive, and it was the earlier stuff that was causing it. But I mean, it doesn't look like it. I don't know how that would work. You'd have
I don't know
like.
if that's exactly. Although I mean, for the Wisconsin data, certainly a lot of the Wisconsin schools would have been a single school, single elementary school, single high school phenotype, for example.
Well, right, and in that case, it's very difficult to imagine you have a crappy elementary school and a great high school. But in some of the other places, you might have, and I'm grasping at straws, but I mean, it's just sort of frustrating.
All right, we'll reconvene in 15 minutes. Thanks again.